Hello, class. The second recording in Module 12 provides an overview of the regulation of protein synthesis. To begin with, it's important to remember that the major stages of protein synthesis are activation, translation, initiation, elongation, termination and release, and folding and processing. Through these steps, eventually you move from primary through secondary to tertiary and finally quaternary structure of proteins that ultimately have their functions in the body. The initial step is activation. It is an energy requiring step to activate the amino acids for the formation of the peptide bond. As the polypeptide continues to develop and the message gets translated, protein synthesis then proceeds. It is translation and initiation as the step that is regulated with regards to protein synthesis, and there are several potential regulatory controls. These controls include amino acid availability, the energy state of the cell, endocrine factors, and initiation factors. In the current recording, we are going to focus on amino acid availability and begin to consider certain aspects of initiation factors, which are the focus of the third recording of the module that is directed at not really an overview, that is directed at specific mechanisms that have been characterized in global protein synthesis. For now, let's consider amino acid availability. In the class, we have talked about the critical role essential amino acids play moving from establishing protein as being the only macronutrient for which there is a requirement, the fact that this requirement is based on the need for indispensable amino acids. They are essential nutrients which must be provided in the diet because we cannot make them, but as you might surmise, they are therefore going to be significant in regulating protein synthesis. The energy state of the cell is intuitively an important point of regulation, given that anabolism, protein synthesis, would be an energy requiring process. Endocrine factors, several shown on this slide, also impact protein synthesis. Insulin is the most significant of those that you see listed on this slide, although much work has been done in considering IGF. Of course, it has many variants. Cortisol, which you would consider as a catabolic nutrient. Glucagon. Both cortisol and glucagon are counterregulatory hormones along with insulin, where insulin will lower blood glucose levels and cortisol and glucagon would elicit an increase in blood glucose levels via gluconeogenesis, glycogenolysis, etc. There are a number of initiation factors, and in this module, we do not have time to move through these in an extensive detail. Fortunately, this is a course to provide a foundation in macronutrient metabolism. So as we move forward, we're going to identify two major initiation factors that have a role in regulating protein synthesis. However, some of the more detailed molecular signals and genetic regulatory factors will be found in more extensive reviews and classes that are more directed at intracellular signaling and metabolic molecular control of protein synthesis. What I'd like to do now is take some time and focus on amino acid availability and set up our discussion for the third recording of this module, which focuses on initiation factors. An abundant supply of amino acids are critical to support protein synthesis and move the muscle into a positive protein balance. This particular article focuses on some of the earlier work that was done that clearly documented the significance of availability of amino acids with regards for skeletal muscle protein synthesis. Let me walk you through this particular figure where the green bars are the data specific to the fasted or the basal state in a group of men, the subjects in these respective trials. The blue bars are the data in response to an intravenous amino acid infusion, and the amino acids infused were both essential and non-essential amino acids. And then finally, in the orange bars, what you have is the response of protein synthesis of relevance to our discussion here, but also protein breakdown, and by subtracting breakdown from synthesis, you get protein balance. So provision of an amino acid mixture intravenously following exercise is illustrated in the orange bars. I'm going to take you back just for a moment to our stable isotope techniques, and in this case, the essential amino acid phenylalanine is the stable isotope that was used to characterize protein synthesis, as well as protein breakdown, and then net protein balance in skeletal muscle of these individuals. The means by which protein synthesis was determined was by incorporation of phenylalanine into the muscle, and protein breakdown characterized by the release of phenylalanine into the circulation. What you'll note right away is that net protein balance is negative in the fasted state. And indeed, you'll see this a couple of times, but it is now well characterized that a positive protein balance, an anabolic state with regards to protein utilization, muscle hypertrophy if you'd like, gains in whole body, lean mass, but also synthesis of required proteins that have a plethora of roles in the body, requires amino acids. When the fasted state is followed by an infusion of an amino acid mixture, you see that there is an increase in protein synthesis. And while there is little to no change in protein breakdown, the increase in synthesis will contribute to the positive protein balance. Exercise is considered an anabolic stimuli for protein synthesis, given that exercise is initially catabolic. You're not building muscle during exercise, but following exercise during that recovery period, there is an improvement in protein balance. However, it will not become positive without the provision of nutrients, and in this case, a mixture of amino acids. So you see that this increased availability of free amino acids to skeletal muscle improve skeletal muscle protein synthesis, will move the muscle into a positive protein balance, And when free amino acids are provided immediately after exercise, the anabolic effect can be maximized. Moving from intravenous infusions, the lab of Wolf with investigators including Violi and in this case Kevin Tipton, moved on to determine whether an oral provision of amino acids would have a similar effect. The Violi study involved endurance exercise, whereas the Tipton study, shown here, involved resistance exercise. Once again, the same stable isotope model was used to evaluate protein synthesis, protein breakdown, and net protein balance. And again, you see that consuming a drink that had no amino acids, again, had no benefit to net muscle protein balance. Again, it's an important point to note that protein balance cannot be positive in the fasted state. However, when these individuals completed their resistance bout and consumed either a mixed amino acid drink or an essential amino acid drink only, for which the nitrogen content of both of these drinks were the same, you can see that although not significant with regards to protein synthesis increasing or changes in protein breakdown, particularly the decrease in protein breakdown with the essential amino acid mixture, that the difference between synthesis and breakdown, i.e. net protein balance, did move from the negative to the positive state for both amino acid mixtures. And indeed, it would appear that the most important aspect of these amino acid mixtures is the provision of essential amino acids. Later, not quite a decade after, can go back and check, well, it was approximately 3 years following, Elizabeth Borshine and colleagues, again in Bob Wolf's laboratory, went back and evaluated this particular approach with more specific direction on the amount of amino acids that would be needed to elicit a positive protein balance and a related increase in muscle protein synthesis. So the changes in net protein balance in response to a mixture of either non-essential and essential amino acids, essential amino acids only, the combination of either the mixed or essential amino acids with carbohydrate were evaluated with regards to skeletal muscle protein balance. And again, looking at the incorporation of phenylalanine into the muscle, as well as the release of a different isotope of phenylalanine from the muscle into the circulation to ultimately calculate net protein balance. The provision of carbohydrate, and we'll see that as we move into the third recording, was to consider the insulin response and the role of insulin in enhancing skeletal muscle protein synthesis and therefore muscle protein balance. The primary outcome of this particular study was that only the essential amino acids are required to get into a positive protein balance in the muscle, and that the provision of carbohydrate, while it appeared to increase protein balance, it was not necessarily additive in this particular scenario. So it is important to appreciate that only the essential amino acids are needed to get an anabolic response in skeletal muscle protein balance, to promote muscle protein synthesis. And again, that verifies that critical role of these indispensable nutrients, the essential amino acids, in the context of supporting body protein, muscle protein health. And, in the context of some of the work that was done by Tipton and later by Stu Phillips and others, moving into post-exercise protein supplementation to enhance the anabolic response to resistance training, consumption orally is certainly as effective as an infusion of the amino acids. And in the context of those of you working or moving into the clinical realm, the infusion would be something more appropriate in a variety of different parenteral nutrition situations, such as critical care or trauma, enteral nutrition for provision of these nutrients into the gut when there is compromised gut function, and the fact that the non-essential amino acids are just that. They are accessible for rates of protein synthesis throughout the body because they are produced. In summary, it is the availability of the essential amino acids that is critical to the maintenance and the stimulation of protein synthesis in vivo.